Good afternoon. Welcome to the educational session, Unlocking New Targets with Molecular Degraders. One of the objectives of the San Antonio Breast Cancer Symposium is to identify research trends in the area, in our area of interest. And we felt it was appropriate to have a session on molecular degraders, which is a new way, or not so new way, of targeting therapeutic vulnerabilities in cancer. So we have assembled 4 presentations on PROTACs, LYTACs, Biparatopic antibodies, and SERDS. And the objective is really to discuss mechanisms, learn about mechanisms of action, strengths, weaknesses, and how to, whether these are good or bad, to target some of the vulnerabilities that we have identified, or we're in the process of identifying, in breast cancer. Each speaker will have 22 minutes, after which we'll have about 3 minutes for questions. Please think about questions for them. I think it's always good to that we honor our speakers with questions. And we'll proceed with the first presentation by Dr. Stephen Hinshaw from the Stanford Cancer Institute, titled PROTACs. Steve?
Great, yeah. All right, let's see. Great, yeah, so first, thanks to Dr. Arteaga for inviting me. It's really nice to be here. And I guess my role here is to convince everybody that targeted protein degradation is really exciting, if you're not already convinced. And specifically, I'll tell you about how we're thinking about small molecules that do this. So what I'll do is give you some big picture concepts that relate to targeted protein degradation and why we think it's so cool. And then at the end of the talk, I'll tell you about how we're thinking about this, this topic in the lab and what we're doing to improve what can be done. So generally, when we think about oncology therapeutics or targeted oncology therapeutics, we're really optimizing for these 2 big picture properties, which are that they should have disease or tissue selectivity, and then they should activate some really powerful type of pharmacology in the targeted tissue. The problem with this kind of concept is that it leaves us with a really narrow sliver of the Venn diagram to go after. Nevertheless, we have we have a huge pharmacopoeia of targeted therapeutics. And just to pick an example from this slide, we have a kinase inhibitor, Saireudee, and other ALK inhibitors that are really useful for going after non-small cell lung cancer because they attack a driver by filling its active site with chemical matter. So essentially we can do this, but the problem is that what we'd like to do is expand our search space for targeted therapeutics. And so we've been thinking about ways we might be able to uncouple these 2 properties of oncology therapeutics so that we can optimize for tissue or disease selectivity and on-target powerful pharmacology separately. And what we call this is division of labor therapeutics. So there are a ton of different ways to sort of activate this concept. I'll tell you about small molecules that do this. The outcome that we're trying to achieve is minimal activity in non-target organs or tissues like the gut, and maximal activity in a disease-selective manner. So how do we think about doing this with small molecules? Of course, the title of my talk is PROTACs, so that's what I'll tell you about. We like thinking about bivalent small molecules, which simply means that these are small molecules that have separate parts that bind to different proteins. So there's a part of the molecule on the left that binds to the estrogen receptor in this example, and a chemical linker connecting it to another small molecule that binds to an E3 ligase, in this case cereblon. And if those of you who were at Bill Kalin's talk this morning will recognize this IMiD here, which it turns out has its own interesting properties that I'll talk about a little bit later in the talk. And of course, this is a really exciting small molecule that degrades estrogen receptor that's being advanced in late-stage clinical trials by Arvinas right now. And I'll give you a few more examples of this kind of a drug later. Okay, so this is what that drug looks like in action. It binds its target in red. This is the estrogen receptor, but it can be any protein that causes disease for which you have a small molecule ligand. And it brings it into proximity with a multi-component ubiquitinating enzyme that then attaches this yellow ubiquitin to the target and marks it for degradation. The substrate recognition component of these ubiquitinating enzymes are generally called E3s. And you might ask, okay, if we have a good ligand for a protein, what's the point of degrading it? We can just feed a patient lots of the compound that inhibits the protein we're after. So I want to point out some advantages of PROTACs and targeted protein degradation in general that should help you get interested in this. So first of all, when we administer a drug, shown in black, its activity or its concentration in tissue or in serum decreases rapidly. And as a result, simple occupancy-based drugs like the ALK inhibitor I showed you are only active so long as they exceed a concentration that produces target inhibition that keeps the active form of the protein you're after below this arbitrarily defined threshold here in this case. So that's very different from what happens with degrader drugs, which may not start out inhibiting a ton of your enzyme that you're after, but after a while, the enzyme is actually cleared from cells. It goes away completely. And that produces a situation where the effects of the drug can long outlast the compound itself in a patient. So that's one thing that's very exciting. We have this durable pharmacology. PROTACs can also produce a gain-of-function mechanism whereby only a small amount of their cellular targets need to be occupied at any given time in order to produce really powerful pharmacologic effects. So here I'm showing you the E3 ligase bound by compound. Only 1 out of 5 E3 ligases here are bound by this, in this case, lenalidomide. And the same is true for target. So you need much less of this compound, at least in principle, in cells to get your pharmacological outcome that you're after. And then finally, these PROTACs can activate, at least in principle, tumor self-destruct circuits. So that actually hasn't been achieved clinically yet, but it's something we're very excited about in the lab. So if we think about an E3 ligase that would normally degrade a cell cycle regulator, a tumor suppressor, something like this, we could in principle hook it up to a neosubstrate that we'd like to degrade in cancer. So the product of an oncogene, a growth factor kinase, something like this, and then create a self-destruction circuit that a tumor cell couldn't escape from. So again, this hasn't been achieved clinically, but it's something we're very excited about. So what's going on in the real world, not the concept world? We essentially have ligands for 2 really good E3 ligases that are commonly used in clinical stage compounds. The 2 E3 ligases are cereblon and VHL, both of which, incidentally, you heard about this morning from Bill Kalin. The cereblon ligands are those IMiDs I talked about. And you might think this is a relatively limited armamentarium for targeted protein degradation. It turns out we can do really well with just these 2 systems. And so I'm showing you now a diagram detailing the vast number of kinases we found we could degrade in the lab. This is an old diagram from around 2020. But you can see we're essentially covering the entire kinome, and we've since gone further. Relevant to this group, here's a degrader for CDK4/6 based on palbociclib, where we have on the left side of the compound a CDK4/6 ligand, chemically linked to one of these IMiDs again. So this brings CDK4 and 6 into proximity with cereblon, resulting in ubiquitin transfer and ultimate degradation. And in the lab, in vitro, when we treat cells with this compound, we see rapid and almost complete degradation of both CDK4 and CDK6. We can characterize the kinetics here. What we can also do is alter this linker component of the compound and produce totally different pharmacology. So we can degrade CDK4 or CDK6 with different kinetics, or we can also make the compound dirty if that's what we want to do. We don't. So what do we learn from all this? So I mentioned several advantages of PROTACs, and I'll just list them here. They have this catalytic mechanism of action, which allows you to get a really profound pharmacologic effect from relatively small amounts of compound, either in tissue or in cells. They also, what I didn't mention is that they often provide a really unique mutational spectrum, a resistance spectrum. So in some cases, resistance to targeted treatments that are in the target protein itself might not provide resistance to the PROTAC due to the different mechanism of action of these compounds. They also can exhibit really interesting polypharmacology. So we've worked on this in the lab where we have a CDK4/6 degrader that also degrades the immune regulatory transcription factor IKZF2, and this might turn out to be clinically important later on. Of course, there are a ton of challenges in developing and designing and developing these PROTACs. I've just listed a few here. maybe the most glaring drawback so far is that in vitro, at least, we see rapid accrual of mutations in the E3 ligases, so not in the targets, but in the effectors of degradation that provide resistance to these compounds. And this sort of circumvents this gain of function catalytic activity I mentioned. So we need new mechanisms to address this resistance. We also need new degradation mechanisms so that we can explore this tissue-specific or disease-specific pharmacology we're after. I didn't mention it, but both VHL and cereblon are widely expressed. And then finally, starting out with these bivalent compounds with long linkers produces a really steep chemistry challenge where you have to really optimize for pharmacokinetics before you can even learn whether you have something that might, in principle, be useful later on. These are unstable compounds, and they don't get into tissue well. So I'm going to tell you about how we thought about attacking these various problems, and for the moment, I'm going to completely throw away the problem of pharmacokinetic optimization and just tell you about really bulky tool compounds, mostly because it's fun, but also because I think it tells us how much is really untapped and still possible. So we were interested in this E3 ligase called KLHDC2, which we know from prior experiments is an extremely active E3 ligase. What's nice about KLHDC2 is that it has a known peptide substrate, and so we simply attach this 7 amino acid peptide substrate via a chemical linker to a broadly active kinase inhibitor and asked whether we could degrade kinases. Sure enough, we could, and we did this cool control experiment where we simply flipped the stereochemistry at this methionine, which is crucial for binding KLHDC2, and we found that in this modified compound, we saw absolutely no kinase degradation, and here I'm just showing you cell viability in the lab, no impact on cell viability, really meaning that very little of this active compound is getting into cells, and all of the pharmacology we're seeing is due to this induced proximity between this new E3 ligase and kinases. So this is exciting to us as a proof of concept. And so what we did is we just designed roughly 40, so here 36 compounds that had the same kind of design with different peptide degrons from the literature. And we found we could activate kinase degradation quite broadly with these new E3 ligases. So this tells us that we can make these tool compounds sort of at will, and we can rationally design them and activate new degradation pathways across the E3 ligase spectrum as long as we know the peptide substrate. Okay, so certainly these peptide-based compounds are tools, but I do want to emphasize the point that once we know we should be going after something, going from proof of concept to drug candidate can be relatively rapid if you have a good chemistry team, as does Arvinas. So here's a really great chemical ligand for KLHDC2 hooked up via a chemical linker to a ligand for the androgen receptor. So this is now a drug candidate being advanced by Arvinas. A similar report from St. Jude just appeared, and they've done completely different chemistry to ligate KLHDC2 and go after bromodomain proteins. So this is really cool and sort of leads me to the point that development in the space is extremely active, and there are many, many clinical trials going on. So I just pulled this from a recent review, and you know, we're hearing from Arvinas this week about early stage clinical trials for combo therapies with their CDK4/6 inhibitor ER degrader combo tests with Pfizer, and then there are a number of other trials going on from notable companies like Chimera and Nurix. And you can see here that sort of a broad array of indications too, so not just breasts. Okay, so we continue to learn about how we can design targeted protein degradation therapeutics using small molecules and how we can do this with PROTACs. I want to spend the remaining time telling you about how we're thinking about moving away from PROTACs, even though it's the title of my talk, into a different class of small molecules we call molecular glues, which are not bivalent, but instead monovalent. And these are really exemplified by old compounds in the literature called cyclosporin and rapamycin. These are monovalent because they don't have two ligands for two different proteins. What they do is bind one protein, and by binding it, create a new interface for a second protein. In the case of cyclosporin, this is the phosphatase calcineurin. In the case of rapamycin, this is the kinase mTOR, and these both turn out to be really useful in immunosuppressives in transplant. Again, we heard about these really interesting IMiD compounds from Bill Kalin, and he actually gave away the punchline here, which is that these bind cereblon, and without any linker and any target ligand, induce the degradation of these really hard-to-drug transcription factors, IKZF1 and 3, and this turns out to be useful for multiple myeloma. So these are very different-looking compounds. How might you think about discovering molecular glues de novo? And what if I told you that not only are these different-looking compounds, they were all discovered as molecular glues after the fact, which is to say that they were found to be clinically useful due to their activity in various clinical contexts, and then much, much later, it was found that they act by bringing proteins together. So how do you optimize for that? How do you find that? We took a relatively crude approach that it turns out, as it turns out, works quite well. So we started with a toy system where we have a good ligand for this transcription factor BRD4, and we used the ligand JQ1 and simply added covalent handles to JQ1. So these are minimal chemical groups that make irreversible bonds with cysteine residues on the surfaces of other proteins. So what happens is that if these modified JQ1 compounds bind BRD4 and then find a cysteine on the surface of an E3 ligase, we should be able to detect that by looking for rapid BRD4 degradation. And indeed, we found that for many compounds, and why the reason I'm showing you this is that we found really surprisingly that small modifications in this covalent acting group produce totally different degradation outcomes. So a cyanoacrylamide group at the top degrades BRD4 via this E3 ligase called DCAF11, and an acrylamide group uses a totally different system that depends on DCAF16. So, this sort of approach doesn't produce drugs. Again, we're dealing with toy compounds, but what it tells us is that we can rapidly go from minimal compounds to binders for E3 ligases, and then presumably be able to develop these later on into bona fide drug candidates, although the path for chemistry is quite long. Okay, so this is sort of the model, but importantly, these are the people that were involved in pursuing this work. I want to finish by asking this question: what else can we do with this chemocentric type of molecular glue discovery? So how can we find these monovalent compounds with really exciting gain-of-function activities de novo? So this is the group that took this on. Z is a chemist in Nathaniel Gray's lab. I would guess for this specific project, he made between 50 and 100 compounds. In general, he's probably made more compounds than anyone else coming through Nathaniel Gray's laboratory, which is really saying something. This is the impressive group that found out how his compounds are working. So instead of going after cysteine, ZZ decided he'd go after anything but cysteine. And so he used this sulfonyl fluoride moiety at the south pole of this ZZ1 compound, which will go after a different set of amino acids. And the attachment position is also interesting. I won't go into that as a detail. Susanna from Ben Ebert's lab used CRISPR to screen for E3 ligases that could produce BRD4 degradation upon treating cells with this compound, and she found two components of an E3 ligase system called CTLH, which doesn't depend on cullins, which is unique to the existing degraders that are available. So here's a cryo-EM structure showing this CTLH complex with the bromo domain of BRD4 in purple right in the middle. And Jakob and Brenda Schulman's lab could derive a really high-resolution, really quite beautiful structure of this recognition complex showing BRD4 in purple, the compound in gray, and then the E3 ligase substrate recognition protein YPEL5 binding both BRD4 and the compound. So this is indeed a molecular glue, and we can see it sticking into YPEL5 here. And if I'm lucky, this video will play as I talk. Let's see. Have to click it again. There we go. So we're going to fade into the high-resolution structure and then look at the molecular interface between BRD4 compound and YPEL5. So this was really exciting to us, not just because we were seeing for the first time a new molecular glue derived from a covalent probe. It turns out that YPEL5 actually has the same fold as cereblon. And not only that, cereblon, the canonical receptor for these IMiD compounds, has the same groove visited by the same chemical matter, although shifted, as you'll see here. Sorry, I should say different chemical matter. And that's important because this groove in cereblon turns out to be hydrophobic, whereas the same groove in YPEL5 turns out to be basic, as you'll see in a moment. And we think that's because YPEL5 normally recognizes phosphotyrosines or nitrosotyrosines. So here's that compound YPEL5 interface that we sort of fortuitously discovered. And also ordered water molecules. So if you're looking closely, you'll say, wait a second, I thought you said sulfonyl fluoride, that's a sulfinic acid. It turns out we'd actually made a prodrug that upon entry into cells gets reduced and hydrolyzed into this sulfinic acid. And once we knew this, we could model all of these intricate interactions between the E3 ligase and the compound, and really think about how we can improve the potency of this compound by adding really tiny chemical modifications. So we added a chloride to make the ZZ2 compound. ZZ2 turns out to be roughly 10-fold as potent as ZZ1, and it's not just because of the electron withdrawing character of this chloride. It really does fill the pocket of YPEL5 much better. So that's essentially what I wanted to tell you there about that story. Let's see if I can advance this here. Yes, okay. So we've got now, we think, a new way, although I've just shown you BRD4, we've got a new way of looking for molecular glues, these monovalent compounds, and our way of doing it is by using covalent chemistry to search for gain of function in cells. So we use cellular systems, sort of small-scale phenotypic screening to look for small molecules that can affect this function. In this case, and for this group, it's degradation, targeted protein degradation. And what's really exciting about this to us is that we can now pre-specify the target we're going after. I'll say more about that in a moment. Molecular structure is super important for this type of work. It's also important to mention that YPEL5 is upregulated in Ewing sarcoma. So now we've got a way of activating this tissue selectivity that I mentioned in the beginning. So we're starting to get towards this idea of sort of separating these two needs we have for tissue selectivity and potent pharmacology. And of course, one of the reasons of mentioning this to this group is that we're going after other diseases, including breast cancer, MRT, and ML. Okay, so this is really the last slide I wanted to show you. So traditionally, when we look for molecular glues, we use a strong binder for cereblon or another E3 ligase, and we just vary this R group here. So we typically don't use covalent chemistry, and we just look for small modifications to an imide that can bring new substrates to cereblon, resulting in their degradation. The problem is we can't pre-specify the substrate, so we can't go after specific diseases. So that's a problem. The other problem is that it turns out cereblon has a very specific subset of proteins it likes to degrade or attach ubiquitin to, and that depends on a structural motif called a G loop in the target. So we're relatively restricted there. Now, what we can do is take ligands for a desired target, say CDK4/6, and look for R groups that induce its degradation, and then go back and look for the ubiquitin ligase. And so this is really what we're focusing on at Stanford now. We're trying to set up a platform, or we are setting up a platform to do this for a number of disease indications, and we're excited about sort of where this will take us in the next 5 years. Great, the timing is fine. I want to thank this group, but more importantly, this really large group of incredibly talented chemists and molecular biologists at Stanford, also collaborators at DFCI and at MPG in Germany, Brenda Schulman's group.
Thank you, Stephen. We have there are 4 microphones on the floor. If there are any questions from the audience, I'll start with one. This rapid development of resistance via mutation in the E3 ligase is interesting to me because that's not something we see in cell culture easily when we use small molecules. So is that a concern? How do you explain that? I mean, if this happens in culture, would you expect that to also happen easily in patients?
Yeah, two ways to answer that question. So first of all, it does appear that E3 ligase mutation or downregulation transcriptionally is happening in vitro when groups are trying these compounds out. At the same time, the trials are going well, and we know targeted therapies work. So there's going to be clinical benefit, but there will be problems to solve too. So we sort of live in the problems to solve area, but that doesn't discount the huge impact this is going to have on patients.
Okay, thank you. Question, please.
Yeah, hi. Very nice talk. I was curious, we often think that with PROTACs, having a covalent binder to the E3 is not a problem.
Right.
I'm curious about your thoughts on the molecular glue. Would having a covalent binder to the E3 in that context be problematic, or do you think it would, you'd still get the catalytic nature of the degradation of the substrate or the target?
Yeah, great question. The covalency, there are kind of a bunch of possibilities here. And the question I thought you were going to ask is it a problem to have covalency on the target side? But that's a separate discussion. Apparently it's fine. You can have a BTK degrader that's clinically active. Yep. We do actually like having covalency on the E3 ligase side. The interesting drawback there is not that it abrogates catalysis, it turns out it probably helps, but it gives you what people in the field call a hook effect. So you can saturate the system relatively easily. I think that'll turn out to be not such a problem in patients because of the rapidly declining concentration of free drug. Oh, it's a great question.
Yeah, thank you. Here on the right.
Yeah, hi. Can you design these degraders for fusion molecules, understanding that there'll be heterogeneity at the fusion site, but less heterogeneity further out?
So the question is about what are the design principles for making heterobivalent compounds? And it seems like you have a question about the linker choice.
Well, for example, you have fusions in a number of tumors that drive the tumor that are characteristic, but the problem in drugging it has been that the actual specific location of the fusion may be varied somewhat, but the intact parts of those two separate genes may still be fairly consistent. So it's a matter of spacing because the actual fusion molecule may be somewhat heterogeneous.
Right. Yeah. So there are a bunch of ways to answer that question. I would say the successful instances of heterobivalent protein degraders thus far have depended on having good ligands already for E3 and for target protein. And in general, the good ligands for target protein hit parts of the target protein that need to be there for that protein to be a driver. So like in a kinase, the active site. And then attaching ubiquitin is just a matter of having something floppy with a lysine, most likely, that'll come in close enough to the E2 to transfer ubiquitin. But there's a whole field, and I'm sure people from Arvinas, maybe you are working on how to deal with androgen receptor, which does have a bunch of different truncation mutations that make it difficult to target.
Yeah. One last quick question from the left, please.
Hi, and can you comment on artificial intelligence guided search of your?
I wish I could, yeah. I mean, we've been having ongoing conversations with drug discovery groups, largely in industry, who claim, and I believe some of their claims are bona fide, accurate, claim to have the ability to design these heterobivalent compounds. What's really important to understand is that linker design is not trivial, and what it's really doing is inducing not just proximity between two proteins, but actual fortuitous protein-protein contacts, and that's probably what makes the most active degraders work well. They're more like glues. And so when companies or academic groups are going after machine learning design technology to get these heterobivalents, what really should be optimized are the fortuitous contacts between the two proteins. And I think some groups are really thinking seriously about that. Our group is mostly focused on sort of chemical discovery campaigns, so we're probably not the right ones to talk to, but we're excited about it for sure. It's coming.
Thank you, Stephen.
Okay, thank you.
Okay, the next presentation is by Weiping Tang from the University of Wisconsin School of Pharmacy. Development of degraders by recruiting tissue-specific lysosome targeting receptors, or LYTACs. Weiping.
All right. Thank you, Carlos, for the invitation and also introduction. So in this session, I'll discuss the lysosome targeting camera or lysosome targeting degraders. Here's my disclosure. So Steve gave a great introduction about targeting protein degradation, so I don't think I need to go through the details. We often just use this simple cartoon to illustrate the principle of induced proximity. Essentially, my lab in the School of Pharmacy at UW Madison is interested in develop bifunctional molecules that can recruit a effector to the targeted protein. Right now, our main focus is on targeted protein degradation, so the effector can be either a E3 ubiquitin ligase or lysosome targeting receptor on the cell surface. The bifunctional molecule can be the PROTAC or proteolysis targeting camera or lysosome targeting cameras. But one can imagine that using the same sort of proximity-inducing principle, you can recruit other types of effectors like kinase, phosphatase, to do different chemistry on the target protein. So far in my group, we have been focusing on degraders, as illustrated here. Today's focus will be more on the LITAC and the related degraders. At the same time, my group has been working on the bifunctional PROTACs, and then also we call this monovalent molecular glue degraders for a number of years. Both of those molecules recruit E3 ubiquitin ligase. The difference is that the PROTAC has the ligand that combines to both target protein and also the E3 ligase, where molecular glue mainly binds to the E3 ligase. The target protein itself often does not really have a well-defined small molecule binding site. So here I sort of just list the major difference between the LITAC and the PROTAC. For the LITAC, we are actually using cell surface lysosome targeting receptors to degrade extracellular proteins. This includes the secrete and also membrane proteins. For PROTAC and monovalent molecular glue degraders, they will induce the degradation of the intracellular protein inside the cell. Right? So PROTAC and molecular glue degraders are very powerful, but they are still limited to the degradation of intracellular proteins. Right? So we're also interested in degrading extracellular proteins, including secrete and membrane proteins, because 40% of the protein are actually secrete or membrane protein. So before doing that, let me just show you briefly how actually nature degrade the extracellular proteins. It turned out that many secrete proteins are glycosylated. This glycosylated protein can interact with cell surface lectins. Lectins are essentially carbohydrate binding proteins. So the carbohydrate portion of this glycosylated protein will bind to the cell surface lectin. And then after this interaction, this entire complex will undergo endocytosis. And then the receptor, or the cell surface receptor, will actually very often separate from the carbohydrate in early endosome. The lysosome targeting receptor, or lectin, will be recycled back to the cell surface, while the glycosylated protein will be pushed to the late endosome and then eventually lysosome for degradation. So this is the natural pathway, right? How for the cells to degrade extracellular protein. And Carolyn Bertozzi's group at Stanford took advantage of one particular actin, in this case, mannose-6-phosphate receptor or chitin-independent mannose-6-phosphate receptor. It's ubiquitously expressed in most cell types. Based on the name, you can imagine this lectin actually binds to mannose-6-phosphate. So that's the natural ligand. This is the first generation lysosome targeting camera, or LITAC. It is a bifunctional molecule with an antibody that can bind to either a cell surface membrane protein target or a secreted target. Then the other end, we have this polymeric glycopeptide that can bind to the cell surface mannose-6-phosphate receptor. So through the formation of this ternary complex, essentially the target protein being degraded, the bifunctional molecule in the middle, and then the mannose-6-phosphate receptor on the other end, the endocytosis will occur, just like the natural process. And then in the early endosome, the mannose-6-phosphate receptor will dissociate from its natural ligand and recycle back to the cell surface. The bifunctional degrader, together with the target protein, will be pushed to the late endosome and also lysosome for degradation. So PROTAC and LITAC clearly are complementary to each other. For PROTAC molecule, it mainly targets the intracellular protein and then uses the ubiquitin proteasome pathway. For LITACs, one can use this strategy to target extracellular proteins, and here are some of the examples. And then the degradation occurred in the lysosome. They also actually share some common features. For example, both PROTAC and LITAC require only a binder in theory, and then this binder does not need to be functional. However, the binder for PROTAC has to be cell permeable or small molecule because they need to get into the cell first. The binder for the LITAC actually can be antibody, peptide, or small molecule. Because the ligand for the cell surface receptor will take the entire thing into the cell. So we were immediately attracted to this strategy, and then we try to expand the scope of the lysosome targeting camera. Okay? So besides mannose-6-phosphate receptor, there are many other cell receptors one can actually use. Many of them have been utilized in drug delivery research for quite a number of years. Okay? One of them is asioglycoprotein receptor, or ASGPR. Okay? So this receptor, ASGPR, is primarily expressed in liver cell and has been used for delivery siRNA. Well, previously, the mannose-6-phosphate receptor is ubiquitously expressed in most cell types. So if one can actually use ASGPR as the lysosome targeting receptor, one can then develop a tissue-specific degrader, which may have a lot of advantage over something that will be ubiquitously degrade many proteins on different tissue and type cells. The ASGPR also has its own natural ligand or well-defined ligand, and it's GalNAc, and one can actually trimerize the GalNAc molecule to make a trimeric ligand. This will bind to the trimeric ASGPR very tightly. So we first tether a biotin molecule to the trigalNAc molecule to drive a trigalNAc motif. And then this bifunction molecule is able to take fluorescent neutroavidin into the cell for degradation. So this neutroavidin will be the example or model system for secret protein. And one can also degrade a membrane protein on the liver cell through the formation of this type of bifunctional ternary complex induced by a bifunction molecule in the middle. And then this bifunction molecule is composed of antibody binder for the target and then trigalNAc molecule that can interact with ASGPR. So we published this work back in 2021. We were not the only one who have this idea. Currently, Bertozzi and David Spiegel actually published the same, similar type of work with us, with his essentially within 3 weeks. So next, I'm going to briefly show you how this works. So this is the bifunctional molecule that can drag the fluorescent neutroavidin into the cell. So indeed, molecule 1 here has a trigalNAc on one end and a biotin on the other end, can induce the uptake of fluorescent neutroavidin, while this control molecule with just a carboxylic acid on the end is not able to do that. And this uptake depends on ASGPR. If we knock down ASGPR using siRNA, we can decrease the uptake significantly. The uptake is also cell type dependent. We try 3 different cell lines with high expression of ASGPR, a medium expression of ASGPR, or no expression of ASGPR. And then you can see the uptake increase while we have more of the ASGPR protein expression. The uptake of the neutroavidin also co-localized with lysosome, right? So this supports the mechanism of lysosome targeting degradation. And then finally, we also conjugated the trigalNAc molecule to an antibody that combines to EGFR. In this case, we use cetuximab. So indeed, this antibody conjugate has a different shift compared to cetuximab itself on the gel. And the antibody trigalNAc conjugate can induce the degradation of EGFR in 2 different cell lines that both express ASGPR. So currently, we're trying to actually degrade a variety of different secreted proteins in the circulation system using this type of bifunctional molecule. Because the liver is actually accounts about 2 to 3% of our body weight. It's the major organ for the breakdown of a variety of different protein. So we're trying to develop small molecules that can selectively target certain secret protein in the circulation system to the liver for degradation. And then this bifunctional molecule can be antibody, peptide, or even small molecule based. And then in the case of small molecule based, there's actually also a chance to have an orally available small molecule to degrade the secret protein. So this is one ongoing direction. So another direction we're going after is degrade protein in the pathogenic tissue. Right? So SGPR can only drag the target protein into liver. Right? In other words, it's limited to the degradation of liver membrane protein. So there are many other tissues. Right? And then indeed, we can actually go back to the drug delivery literature, and then there are actually a number of receptors have been utilized for drug delivery. The first one we pick is a folate receptor. This is the folate ligand, the small molecule ligand for folate receptor, and the folate receptor is overexpressed in a variety of different cancers. And then folate conjugated PROTAC has also actually been developed. Okay. So our proposal is to conjugate the folate ligand to an antibody that combines to a cell surface protein. And then this bifunctional molecule hopefully will induce the degradation of the target protein selectively on the cancer cell over other cell types. So because we also have tools, right, two compounds for mannose-6-phosphate receptor and ASPR, so we just compared, right, these three different conjugates. Okay. So we conjugate the folate to cetuximab and compared it with ASPR ligand conjugate and mannose-6-phosphate receptor ligand conjugate, and then this is the unconjugated antibody. And then in this case, we actually observed significantly more potent degradation of membrane protein, EGFR, compared to two others have been reported previously. So this may actually contribute to the high binding between the folate receptor and the folate ligand, which is in the picomolar range. So the scope actually quite broad. We tested a number of different cell lines. We were able to degrade the PD-L1 in this case in a number of different cell lines, including triple-negative breast cancer cell line. And we also did a colocalization experiment. The membrane protein indeed is dragged into the lysosome for degradation. So we also compared the degradation of EGFR and PD-L1 in cancer cell versus normal cell. And then in most cases, we observe much more significant degradation of either EGFR or PD-L1 in the cancer cell that overexpress folate receptor compared to normal cells. So next, we decide to move on to the in vivo study, and then the first thing we do is test the PK of the antibody conjugate, because we actually modify the antibody by attaching a ligand, in this case, folate ligand. Well, we found that this conjugation did not really actually change the PK of the antibody that much. Okay, so the antibody conjugate and an antibody have very similar PK. So next, we test the in vivo degradation of PD-L1. Then by IHC and Western blot, we indeed actually observed degradation of PD-L1 in vivo. And then also we observed increase of CD8 plus infiltration cell in the degrader case compared to just the antibody alone. And then we also isolated other tissue and did not really see a significant change of the morphology and also the PD-L1 level in other tissue. So in vivo, we also were able to achieve tumor selective degradation of PD-L1. And then we tested the efficacy of the PD-L1 degrader in 3 different syngeneic mouse models, B16F10, CT26, and also MARC1 head and neck cancer model. In all 3 cases, we observed more dramatic anti-tumor efficacy for the degrader group in red compared to the antibody group in blue. And in all 3 cases, we did not observe a change of the body weight of the mice. So also selected some mechanistic studies in this to highlight in these slides. So we treat the cell with the degrader and also co-treat the cell with the degrader together with its nature ligand folate. Okay. And we found that using high concentration of the folate ligand, we can actually abolish the degradation of the PD-L1 and also EGFR induced by the antibody conjugate. Okay. So if you look at the concentration of the degrader, right, they're in the 10 nanomolar range. Okay. While the folate ligand, we actually need to use almost 100,000 to millions fold, right, to abolish the degradation. So this actually is also interesting. Okay. So in this field, right, there's always concerns of competition with the natural ligand, because we have a micromolar concentration of the folate in the serum. Okay. So our experiment here showed that this competition actually may not be a concern. We have to use really minimal concentration of the folate to abolish the degradation induced by nanomolar concentration of the antibody conjugate. The degradation cannot be abolished by proteasome inhibitor, and then if we knock out Rep7, which is the essential protein in the endosome, in early endosome to late endosome formation. So in this case, we did not see the abolishment. In this case, we see the abolishment. So these all are consistent with the proposed lysosome sort of mediated degradation mechanism. So in summary, we have developed tissue-selective degraders that recruit SPR and folate receptor for secreted and membrane proteins. And throughout the process, we also actually develop a site-specific labeling method for the synthesis of antibody conjugates. And we're currently using this technology to develop degraders of other disease-relevant proteins. But we're actually more interested in discovering a new type of tissue-selective lysosome targeting receptors as a platform to degrade a variety of different proteins in different cell types. So this is a review paper we published in 2023. By that time, actually a number of lysosome targeting receptors have been validated for the development of LITEX, including integrin, transferrin, and recently RP1. So this will be my, not sure why it doesn't really show up here. So I've been often asked, right, how do you compare, right, the degrader with the antibody? Right? Other targets you can actually go after what antibody cannot do, because for many of the cell membrane protein, you can use or secreted protein, you can use antibody to target them. So we have been thinking about this for quite a while, and then have some different classes. Right now, we're actually focusing on some of the multifunctional proteins that will be challenging to address using antibody, because one can only bind to a specific functional site of the antibody. Or this degrader, we only need to have a binder that binds to somewhere around the protein and then drag the entire thing for degradation. All right, with that, I'm going to thank the group who actually worked on this, and then also the funding agency to support our research.
Thank you, Weiping. Questions from the audience?
Very nice. I'm just wondering about receptor density and the spatial degradation. So, for instance, you have to be in quite close proximity, obviously, to get your target and the two molecules together, right? So, and proteins are differentially presented at different parts of the membrane. So, do you actually find that there's differences in the sensitivity based upon the fluid dynamics or, RAF formation and things like that with these class of molecules?
Yeah, great question. So we believe all molecules are dynamic, even molecules on the membrane, like a membrane protein, right, EGFR, PD-L1, they are all actually dynamic. They actually can flow, right, quite dynamically. So I don't think we need to worry about their intrinsic sort of location in this case. But we did actually also examine how the linker length, right, will impact the degradation efficiency. So it really actually depends on the molecule we use. Yeah. Okay? So if we use antibody as the binder for a target protein, the antibody itself is huge. Mm-hmm. So we have to use a very long linker to induce the degradation. Mm-hmm. The short linker just don't work. Yeah. And then very likely the short linker just buried within the antibody. But if we use a small molecule binder for the target protein, the linker doesn't need to be too long. Yeah. So we're moving very close to the PROTAC space. The linker is around 10 to 20 atoms, and we're trying to optimize that when we have a small molecule ligand for both ends.
Yeah. And then very quickly, the second question then is, have you had I could probably read your papers and ask this, but an appreciation to, what level of expression of the various receptors that you need in order to be able to accomplish a useful turnover using the LITEC approach?
Yeah. Great question. So we often actually try to use receptors that are overexpressed in certain tissue, right? And then we try to correlate the degradation with the expression level of the target protein and also the receptor. very often we find the degradation is related to both, or sometimes just a ratio of these two. So you have to have an ideal ratio. If you have too much of a target or too little of a target, you just don't really see very efficient degradation, right? Too much, too little compared to the receptor. But the receptor itself is often overexpressed. We have not really looked at the a low expression receptor.
Let me ask you about another variable, which is the antibody. So I can think of antibodies that recognize the target, also, but are agonistic. They induce tumor growth. Would that be a problem?
Antibody itself.
Yeah, antibodies can be agonistic and induce tumor growth. Is that a problem for the generation of a LITAC? Because we don't want the tumor to grow, right?
So what's the mechanism for the induced growth?
I don't know, but they behave like ligands. The question is because I am interested in the target and I have some antibodies and I want to know which is the one I'm going to bring to you. I want to bring you the right antibody, not the one that induces tumor growth.
Yeah, this is interesting because if the induction of tumor growth is induced by the target itself, right?
It is,
You
yes.
block the function of the target and then the cell just try to express more, right? Then in that case, degrader may actually be better.
Okay.
And then that's why actually, PROTACs develop EGFR for that reason.
Okay.
So degradation can be,
Let's follow that because I'm thinking about what antibody to bring to you. So thank you very much. Okay, the next presentation is by Saireudee, Chaturantabut, titled Biparatopic Antibodies Targeting FGFR2.
So thank you, Dr. Arteaga, for the invitation and for the opportunity for me to come to here and share with you today about our ongoing work on biparatopic antibodies. And particularly, I'll be sharing with you a specific target targeting FGFR2. This is my disclosure slide. And as the title suggests, I'll be focusing on the specific type of antibodies called biparatopic antibodies, which we think it's a promising strategies to target oncogenic receptor for degradation. So what is biparatopic antibodies? Biparatopic antibody is a subset of bispecific antibody which bind to two different epitopes. And if these two epitopes are on the same antigen, we call them biparatopic antibodies. There are multiple types of biparatopic antibodies, multiple formats. What we selected for here is IgG format because of the similarity to the endogenous antibodies, and which we hope that it will reduce immunogenicity and also can possess the desired properties such as a long half-life. So the technology that we use to synthesize these antibodies is called DuoBot technology. And this technology is based upon creating the mutations in the Fc region of the antibodies. And upon introducing the reducing environment, this mutation will favor heterodimer and will form bispecific or biparatopic antibodies. So as I mentioned, I'll be focusing on the FGFR2 as the target as an example today. And oncogenic FGFR2 that's commonly found in cancer, there are multiple types of alterations, including amplification, mutation, and fusion. And oncogenic FGFR2 is implicated in multiple cancer types, for example, lung cancer, intrahepatic cholangiocarcinoma, or ICC, gastric cancer, as well as breast cancer with FGFR2 amplification. So the first story I'll share to you today is to focus on ICC, which is driven by FGFR2 fusion, and the fusion partner is here attached in the C-terminus domain. And the reason we chose this FGFR2 fusion is because, one, because it's clinically needed, but also two, these fusion partners, which is a strong driver of oncogenicity, is really setting up a high bar for us to hopefully be able to inhibit the growth of these tumors. So FGFR2 fusion is clinically validated, and with that, multiple companies have been developing inhibitors, including pemigatinib, infraganib, and futibatinib, which are all FDA approved, has been shown to have good activities. Nevertheless, the activity of these compound result in an overall response rate lower than 45% and with progression-free survival of less than 9 months. And so compared to other RTK inhibitors, their activities are still pretty limited. So what we think might be the challenge includes, one, the emergence of resistance, and also the on-target but off-tumor effects, in this case by FGFR1 inhibition. And this is due to the high homology between FGFR1, 2, and 3, which make it difficult to target FGFR2 specifically. So with that, we hypothesized that by targeting the extracellular domain of FGFR2 with biparatopic antibodies, it would increase selectivity, potency, and also overcome FGFR inhibitors' acquired resistance. So to generate biparatopic antibodies, we first need to select the parental antibodies. And here we identified 6 different parental antibodies from patent literature. These are therapeutic antibodies with various epitopes across the ECD domain, including D1, D2, and D3. And important to note that D2 and D3 are important and necessary for ligand binding and ligand-induced dimerization and activation. So we look at the activity of these 6 parental antibodies in FGFR2 fusion-expressing BAF3 cell system in the presence and absence of ligand. And the reason we look at both in the presence and absence of ligand because we don't know in the patient tumors whether it is dependent or independent of the FGF ligand. And so we want antibody that work in both settings. So here, in the presence of ligand, we see that antibody that bind to the ligand binding domain, C, D, E, and F, can block ligand-induced growth, which is expected. However, in the absence of ligand, only antibody F have some activities. Antibody B, D, and E have minimal activity, while antibody A and C are actually act as an agonist. So it might be interesting to note as well that antibody E here is a parental antibody for antibody bemarituzumab, which is the ADCC enhanced version currently being used in the FGFR2 positive HER2 negative gastric cancer patient treatment. And so overall, the data here suggests that the parental antibody activity is fairly limited. And so with that, we move on to generate biparatopic antibody, creating a combination of all 6 by 6, and that leads to the 15 different biparatopic antibodies. We then tested the affinity of these antibodies, and we found that 80% or 12 out of 15 biparatopic antibody generated has enhanced binding affinity compared to their parental antibodies over 10-fold. And the 3 biparatopic antibody that have the lower affinity shown here, AE, CD, and EF. These antibodies either bind to the same binding domain or bind to 2 different domains that are likely to be furthest apart. So we think that these antibodies might be using just one binding arm and therefore might result in the lower binding affinities. We tested the ability to inhibit FGFR2 fusion-driven cells growth here in the viability assays in the absence and presence of ligand, and we found that 50% and about 80% of biparatopic antibody outperformed their parental antibodies in the absence and presence of FGF10 respectively. And with this, we selected the top 2 biparatopic antibody with the highest activity in both settings, BC and BD combinations, to move forward to characterize further. We next measure the binding avidity, which is the combinations of all binding affinities of a single molecule using the acoustic force spectrometry. Briefly, we have these antigen-positive cells at the bottom, and the antibody will be conjugated to the beads, which is labeled by, with fluorescence. And upon applying the force, acoustic force from 0 to 1,000 piconewton, this antibody will slowly being removed according to its avidity. So shown here on the right, our top biparatopic antibody, BC and BD, show the highest avidity compared to parental antibodies and controls. And it's interesting to note that other biparatopic antibody, A, B, and DE, which show high affinity but lower avidity here, suggesting that the activity of the biparatopic antibody might be better predicted, represented by the binding avidity rather than affinity. So we further show as well that these biparatopic antibodies are able to inhibit transformation of FGFR2 fusion-driven transformation. Here we use 3T3 system at a dose-dependent manner compared to their parental antibodies and controls. We move on to test in vivo using intrahepatic cholangiocarcinoma cell line ICC137 xenografts model. And while we see limited impact of the parental antibodies on this tumor growth, we saw significantly growth inhibition in these biparatopic antibodies at 2 concentration tested. So we want to see what's the molecular mechanism of these antibody by isolating the tumors, and we look at different protein expression. And here we surprisingly found that the FGFR2 receptor is dramatically downregulated. And so we hypothesized that this may be due to the antibody inducing internalization and degradation of the receptor. So to prove this, we label parental antibody and biparatopic antibody at the FC region with the Fab fragment conjugated to the pH-sensitive dye. And this dye, upon being internalized and being exposed to lysosomal environment, it will fluoresce. And so the signal that we detect here will suggest the lysosomal mediated degradation. So what we found was that the biparatopic antibody that we selected show a much higher signal compared to the parental antibodies and controls, suggesting that indeed these biparatopic antibody are able to induce strong internalization and receptor-mediated degradation. So far the model that we have in mind is that in fusion-driven tumors, the receptor will spontaneously dimerize because of the fusion partners, and that leads to the pathway activation and tumor growth. Upon addition of the biparatopic antibody, this antibody will recruit receptors to form this high complex oligomer, which then trigger internalization and degradation through the lysosome pathway, and that would lead to tumor growth inhibition. So with that model in mind, we asked whether our antibody would be active against various kinase domain mutations. And there are multiple kinase domain mutations that were discovered at progression in patient tumors that has been treated with antibody, that has been treated with FGFR inhibitors. So that include the residue V565 gatekeeper mutations, as well as N550 molecular brake mutations. So since given the location of the kinase domain mutation, which is intracellular, and the binding site of the biparatopic antibody extracellularly, so we hypothesized that our antibody should remain active. And indeed, we created this model with VF and VI mutation, and these mutants are resistant to the FGFR inhibitors. However, our antibody are able to inhibit the growth, induce internalization, and as well as receptor degradation. And similarly, we show in other mutations as well that these biparatopic antibodies are still active at inhibiting growth of kinase domain mutations through receptor degradation. Finally, we asked whether our biparatopic antibody are active in the context of FGFR2 wild type, and particularly in the amplification setting. FGFR2 amplification has been found in multiple types of cancer, including breast cancer and gastric cancers, which I'll be using as an example today. It's been shown that about 10 to 15% of gastric cancers are due to the FGFR2 amplifications, as shown here in the CTGA cohort. And interestingly, FGFR2 expression in these gastric cancers are heterogeneous, and these really lead to a variable response to different therapies. So to test whether our antibodies are active against the wild-type FGFR2, we created this model FGFR2 overexpressing BAF3 cell system, and we show that majority of the biparatopic antibodies are active, and particularly our top 2 biparatopic antibody remain pretty active against this wild-type. We also tested in SNU16 with FGFR2 amplification gastric cancer cells, and we found that our biparatopic antibody are able to downregulate the receptor both in the absence and presence of the ligand. So given the high specificity and the high internalization capability of our biparatopic antibody, we really think that it's going to be a good candidate to develop antibody-drug conjugates to enhance its potency. And another reason that biparatopic antibody could be interesting in developing the ADCs is because it could increase the payload delivery due to the high antibody receptor ratio with the mode of binding that I've shown earlier on the right, which gives a 1-to-1 binding versus 1-to-2 compared to the traditional bivalent antibody. So with that, we generated multiple payload linker combinations, both non-cleavable and cleavable linker, and this payload linker combination has already been used in the clinics. We tested these different compounds in the SNO16 xenografts model with FGFR2 amplification, and upon a single dose treatment, these tumors all regress pretty continuously and sustainably, which is encouraging. But importantly, the heterogeneity that I mentioned in terms of FGFR2 expression in gastric cancers, we really hope that by using the cleavable linker payload, we would be able to enhance or exhibit the bystander effects, which will kill not just the positive cells, but the negative cells that are surrounding it. So to test this, we engineer the cell lines with FGFR2 negative and FGFR2 positive cells to express GFP and RFP. And this way, we will be able to co-culture them and track their growth individually or in the admix context. So when we treated these cells with the MMAE, which is a cleavable linker to conjugate it to the biparatopic antibodies, we see that in FGFR2 positive cells, it's shown the effective growth inhibition. In the FGFR2 negative cells, these cells can only be inhibited only in the admix context, or in other words, in the presence of both FGFR2 positive and FGFR2 negative cells. But these compounds will not inhibit the growth into FGFR2 negative cells in isolation. So this really suggests that the biparatopic antibody ADCs can inhibit growth of these cancers specifically in the FGFR2 positive population, but also exhibit the bystander effects, killing cells that are cancer cells that are surrounding the positive cells. So this is really encouraging to us, and hopefully that the molecule like this will be helpful in targeting receptor expression that is heterogeneous, such as in the gastric cancer. So to summarize, I've shared with you that we created these biparatopic antibodies that has distinct pharmacology compared to the parental antibodies, which is not predicted by the antibody mixtures. And through lysosomal mediated degradation, and biparatopic antibody has downregulated FGFR2, which then result in the growth inhibition in vitro and in vivo. Biparatopic antibodies are active against kinase domains mutations, as well as ECD oncogenic mutations, which I didn't have the time to show today, but also is active against FGFR2 amplify wild-type cancer cell lines, and ADCs are currently under development. Just the last part, as I mentioned before, our top biparatopic antibody consists of 2 binding arms. So one binds to the D1 domain, and the second arm binds to the D2 domain. It's important to note that D1 domain is a flexible autoinhibitory domain. So this pairing happened to be perfect to cross-link these receptors and activate trigger the internalization and receptor degradation. But it's important to note that not all biparatopic antibody will bind in this mode of binding, and also not all biparatopic antibodies would result in a strong antagonistic molecule. And really, the key here is the epitope selection. It's a good determinant of the biparatopic antibody activity. So with that, we want to look into this more in a more systematic way. So currently, we are in collaboration with the Center for Therapeutics at the Broad Institute. We're developing a ribosomal display to generate the nanobodies from a large unbiased library, which will select the binders and then make the combinations to generate biparatopic antibodies. And with that, we will characterize the functionality and then the epitopes of these biparatopic antibodies to be able to understand the correlation or a better way to predict and engineer these antibodies to be active in our as we need it in different against different RTKs. So with that, I'd like to thank the Sellers Lab, Bill Sellers, who's leading the team, and other members of the lab who contribute to this work, past and present, our collaborator, and our funding sources. So thank you for attention.
Thank you, Gal. Questions from the audience? So maybe I can start. So FGFR2, is an oncogene in triple-negative breast cancer. It's amplified. It's also mutated, and it's also the target of fusions. Not a high frequency, but they happen, and it's true oncogene there. So I know of only one FGFR2 selective small molecule. It's published for a company called Relay. The dose-limiting toxicity, I think, was diarrhea. That's the one FGFR2 specific toxicity.
Relay, yep.
Can you speculate as to whether this approach would be different in terms of that toxicity? Clearly, it will change the scheduling of the drug, right? You're giving a big, small molecule, versus a small molecule, different. The pharmacology will be different. But would you expect the toxicity to be different or the same?
Right, yes. So I think that's great. There's a new compound from Relay Company, 4008, which are able to target FGFR2 more specifically. It's not currently approved, but definitely really promising. We actually work really closely to one of the labs who have been testing the compound. And we still found that one of the limiting factor is still the resistant that still eventually emerge.
To resistance to what? To your drug or to Relay?
To the Relay.
Okay.
So I think that would be maybe one benefit in terms of what our antibody could complement in terms of different treatment strategies. But of course, I think the FGFR1 on target but off tumor effects will be much reduced with that compound, and the toxicity will be reduced. So I think in terms of toxicity will be reduced from the small molecule side, but for the antibody, its toxicity is definitely far less in general.
Far less.
Far less, yeah.
Huh, okay. We'll stay tuned. Please.
So I was interested in the activity on the kinase domain mutations, because I was curious, do you know whether those still have to signal as dimers, or whether those FGFR2 kinase mutant receptors still dimerize? Because I understood that the dimerization of the receptor was part of the way you had the enhanced affinity with your...
Right, yeah, no, that's a great question. I think there are multiple type of mutations, and there's definitely some that are at the ATP binding domain, and that's basically block the binding of the drugs, and that's why it's resistant to particular drug. But definitely the dimerization is still there. So I think, and we tested in our antibody against those, yeah.
And can you remind us, how do you explain the bystander effect? I was not clear on that.
Right, yeah, so and it's more like for the ADC story. What we're trying to do is really to target the FGFR2, in this case, positive cells, but also the surrounding cells, which is the cells that are in proximity, which is FGFR2 negative, and we try to be able to kill those cells as well. So we actually I can share a little bit. We also are doing the in vivo experiments to confirm that by implanting the xenograft tumors in both sides, one which is the negative cells alone, and the other side is the admix of positive and negative cells. So our hope is that the ADCs would be able to kill the tumor on the side that with the admix, positive and negative cells, but not on the other side, which is the negative cells alone, which is kind of mimic, other organs in our body with the low expression of FGFR2. So we're aiming to target the surrounding, like nearby.
Weiping.
Yeah, just a technical question. You mentioned two epitopes of, so it must be two H and two light chain. And how do you actually put them into one, or you do chemical engineering?
Yeah, so we use the dual body technology. So we essentially create the mutation in the FC region. And so we create parental antibodies separately. And this parental antibody contains specific complementary mutations. And then we combine them and then introduce the reducing environment. And that forced the the complementary mutations to swap and prefer the heterodimer. So it's pretty tricky process, and it took us a while to optimize. But yes, it's definitely a challenge.
Rachel?
What do you know about the brain penetration, the blood-brain barrier or CNS?
Yeah, I think currently it's still quite limited. Yeah, unfortunately.
Thank you very much. Okay, the last presentation is by Dr. Rinath Jeselsohn, titled Latest on Selective Estrogen Receptor Degraders, SERDS.
Yeah. Yeah. All right. So thank you. And my title is latest on selective estrogen receptor degraders, or SERDS. However, I will start with a history on the SERDS. These are my disclosures. So the first pure estrogen receptor antagonist, which is the ICI 164384, was actually discovered in 1987 by Weiping and Bowler. And as you can see here, this was a derivative of estradiol with a bulky side chain. And later on, fulvestrant was discovered, also a pure ER antagonist, and these were considered pure ER antagonists because they don't have agonist activity in the uterus or other tissues. And this was shown to be a more potent antagonist, as well as the ER degradation properties of these drugs were characterized at that time. And later on, another ER antagonist with ER degrader activity, GW5638, which is a derivative of tamoxifen, was discovered. And then finally, in 2002, the first ER degrader, or fulvestrant, was approved for the treatment of metastatic disease post-progression on first-line treatment with an endocrine therapy. Now, already at that time, it was already recognized that the PKs of fulvestrant are limited, really limiting the activity of this drug. And furthermore, in 2013, the ER mutations were recognized as a key mechanism of resistance to endocrine therapy, and they have relative resistance to fulvestrant. So really, this really prompted the continued development of new SERDs. So the first generation of oral SERDs had an acrylic acid side chain, and these SERDs were basically mostly, their development has been discontinued. Last year, the first oral SERD, which has a basic side chain, was approved for the treatment of metastatic ESR1 mutant breast cancer patients who had progression prior treatment with endocrine therapy. And currently, in addition to elacestrant, there are a number of other oral SERDs with basic side chains that are in advanced phases of clinical trials, looking at them both as single agents and in combination. So let's just go back to some of the very basics on the estrogen receptor. So the estrogen receptor is a nuclear hormone receptor, and when estradiol is bound to ER, it basically activates the receptor. The receptor is translocated to the nucleus, where it binds to chromatin either directly or tethered to other transcription factors, and it's really the binding of the co-activators versus co-repressors, so this balance between them that really dictates the transcriptional activity of the estrogen receptor. Now this is the diagram of the estrogen receptor protein, and as you can see here to the left in the N-terminal is the AF1, or ligand independent transactivation domain, which really is intrinsically disordered, and still we know very little about AF1. On the other hand, to the right is the N-terminal AF2, or ligand-dependent transactivation domain, or ligand-binding domain, which we know more about this domain, as this domain, the structure of this domain has also been solved. Shown here is the figure of the crystal structure of the ligand binding domain. And you can see here that it's composed of 12 helices that make up a three-layer composition of an internal layer and 2 outer layers. And it's really this composition that allows for this large hydrophobic cavity, which is the ligand binding pocket. And when estradiol is bound to the ligand binding domain, it sits in this pocket. And really, what's important is that when estradiol is bound to the ligand binding domain, helix 12 sits above this pocket. Although it doesn't touch the estradiol, it forms a lid to the cavity. And in this position, the helix 12 hydrophobic surface is facing towards the cavity. And this specific positioning of helix 12 is really what allows a competent AF2 cleft, and that is what allows for co-activator binding, which is necessary for the agonistic activity of the estrogen receptor. On the other hand, when tamoxifen is bound to the ligand binding domain, it also sits in the ligand binding pocket, similar to estradiol. However, because it has a side chain, a bulky side chain, there's no room for tamoxifen in this cavity, and that leads to the protrusion of helix 12. And this position of helix 12 is now interfering with the AF2 cleft and really leading to an antagonistic activity. And really this is really the essence of the antagonistic activity of all the antagonistic ligands. So what about fulvestrant? So the structure of fulvestrant bound to the ligand binding domain has not been, the crystal structure has not been solved. However, the crystal structure of GW5638, which I alluded to before, bound to the ligand binding domain, was solved by Jeffrey Green, and shown here are orthogonal views of both of GW and superimposed with tamoxifen bound to the ligand binding domain. And the cylinders basically represent the helix 12. The blue cylinder is helix 12 when tamoxifen is bound to the ligand binding domain, and the yellow cylinder is GW bound to the ligand binding domain. And as you can see here clearly is that with these different antagonists, the positioning of the helix 12 is very different, already signifying that they're doing different things and their antagonistic activity may be different. Furthermore, when we look at a higher magnification at the ligand binding domain with these two ligands, you can see that not only does GW interfere with the co-activator binding, but it also leads to repositioning of the hydrophobic surface of the helix 12 in a manner that now the helix 12 hydrophobic surface is now facing towards outside. And it's really this facing of the hydrophobic surface that leads to the instability of the estrogen receptor, and that leads to further degradation of estrogen receptor by the ubiquitin proteasomal system. So furthermore, if you look at the surface hydrophobicity of the different ligands, you can see really that level of surface hydrophobicity is really different, and that's really what dictates the different or the spectrum of ER degradation with these ER antagonists. As you can see here, ICI or a SERD has the highest surface hydrophobicity, while tamoxifen has the lowest ER hydrophobicity. And when you look at the correlation between surface hydrophobicity and ER degradation or ER protein level, there's clear correlation. I want to point out that tamoxifen, for instance, has the lowest surface hydrophobicity and actually causes stabilization of the ER protein, as you can see here in this Western blot. And furthermore, to really validate that it's the it's the surface hydrophobicity that is really critical for the ER degradation, Jeffrey Green's group generated ER mutations in which the hydrophobic amino acids were replaced by hydrophilic amino acids, as you can see here in this Western blot to the right. And you can see that when doing that switch, you really lose ER degradation. So I've shown you that, SERDs have two modes of action. They alter the helix 12 conformation, blocking the AF2 cleft, and that prevents co-activator recruitment, resulting in antagonistic activity. The helix 12 also is altered in response to SERDs, and that increases the surface hydrophobicity of the ligand binding domain, which is associated with instability and increased degradation. A recent work by Ben Ebert's group at Dana-Farber had shown that this proteasomal degradation likely starts within the nucleus itself, and by doing CRISPR screens, they've actually identified the E3 ligases that are responsible for the ER degradation in most of the SERDs. There are also a number of studies that have shown that SERDs decrease the nuclear mobility of ER, and this effect is associated with the antagonistic activity of SERDs. However, it's still not clear if this effect is really driving the activity of the drug, or is this actually a consequence. So there are clearly two modes of action of fulvestrant, and one question that comes up in the field is really, is ER degradation the determinant of the inhibition of ER-mediated transcription and the anti-tumor activity of SERDs? And Donald McDonald, who's sitting right here from the University of Duke, has done work to really answer this question by taking two approaches, both by inhibiting the degradation of ER by pharmacologically with a proteasomal inhibitor, or by overexpressing ER, leading to saturation of ER degradation by fulvestrant. And in both of these approaches, he showed that really the ER degradation is not essential for the ER inhibition activity of fulvestrant, suggesting that really the degradation is not the determinant of the activity of the drug. So what are some of the limitations of fulvestrant, and why do we need next generation SERDs? So fulvestrant is an intramuscular injection, and for many patients this is very uncomfortable. I mentioned that it has poor bioavailability and pharmacokinetics that really limit the full potential of fulvestrant. And also the ESR1 mutations confer relative resistance to fulvestrant, and this has been shown by biophysical studies that have shown that in the presence of the ER mutations, the affinity of fulvestrant is decreased, as well as the antagonistic potency of fulvestrant. And we have shown in functional studies that the presence of the ER mutations decreases the ER degradation, as you can see to the right on the top, and also decreases the effect on transcription. So why are the ER mutations important? We know that they lead to stabilization of helix 12 in an active conformation, leading to the constitutive activity of ER and aromatase inhibitor resistance. They also importantly have neomorphic properties that are strong drivers of metastases. And also, although they are uncommon in primary disease, they are highly prevalent in metastatic disease after the acquisition of resistance to endocrine treatment. And in some of the studies, as shown here for an example in the PACE study, these mutations are found in more than 50% of patients with metastatic ER-positive breast cancer. It's important to highlight that the ESR1 mutations are really a mechanism of endocrine resistance that is ligand independent but ER dependent. So what clinical evidence do we have for the relative resistance to fulvestrant as monotherapy in the presence of the ER mutations? This is a retrospective analysis of the PALOMA-3 trial, looking only at the fulvestrant arm and looking at the subgroups of patients who either had wild-type ER, as in blue, or mutant ER and in red. And as you can see here clearly, patients who have an ER mutation do worse with fulvestrant alone. In addition, very similarly, similar results were seen in the MONARCH 2 exploratory retrospective analysis. So elacestrant is the first oral SERD to be approved, and only oral SERD that is currently approved. It was approved for ER-positive, ESR1 mutant metastatic breast cancer after at least one line of endocrine therapy. And what do we know about elacestrant? So elacestrant has a unique pharmacology. At lower concentrations, it has agonistic activity, and at higher concentrations, it has antagonistic activity. There are a number of preclinical studies showing that it has superior activity compared to fulvestrant in models of endocrine resistance. And also the crystal structure of elacestrant bound to the ligand binding domain has been solved, and it has shown that it has a unique position in the ligand binding pocket and really unique interactions with helix 12, leading to potent antagonistic conformation. It is the EMERALD phase 3 trial that eventually led to the approval of elacestrant. In this trial, patients who had progressed on prior treatment with endocrine treatment and a CDK4/6 inhibitor were randomized to either elacestrant alone or physician's choice of endocrine therapy. This study had 2 primary endpoints, PFS in all patients and PFS in ESR1 mutant patients. And as you can see here, in the first primary endpoint was a positive study, although in all patients, the absolute difference in PFS with elacestrant was not clinically meaningful. However, in the presence of the ER mutation, the difference was more significant. However, despite this being a positive study, it's important to highlight that about 30% of patients with an ESR1 mutation had a very rapid progression with a single agent elacestrant. And this really leads to the question, the need to identify the subgroups of patients and the need for biomarkers in addition to the ESR1 mutations to identify those patients that really benefit from a single agent oral SERD. So the exploratory analysis of the EMERALD trial looking at different subgroups has started to help us try to understand who these patients are. And in this analysis, it was shown that longer duration on prior treatment with a CDK4/6 inhibitor is associated with improved benefit from elacestrant in patients with an ESR1 mutation. As you can see here in this graph, the blue represents patients who had a longer duration on CDK4/6 inhibitor and endocrine treatment. And what about patients with PIK3CA mutations and an ESR1 mutation? Since PIK3CA is a very common mutation in ER-positive breast cancer, as you can see here on the top, patients with an ESR1 mutation and PIK3CA mutations still do benefit from elacestrant compared to standard of care endocrine therapy. But as you can see here on the bottom, patients without a PIK3CA mutation versus those with a PIK3CA mutation do better on a monotherapy elacestrant. So in addition to elacestrant, as I mentioned, there are several other oral SERDs in advanced phases of clinical development. Shown here are some of the phase 2 and phase 3 trials of single agent SERDs. I also added here is the Veritech study, which is looking at the oral ER PROTAC. And these are all looking at single-agent SERDs as second-line treatment in metastatic disease. As I've shown you, EMERALD was reported and was a positive study. Some of these other studies were already reported. And this morning we heard results from the EMERALD 3 trial, which is a phase 3 trial looking at imlunestrant versus standard of care endocrine therapy versus imlunestrant plus abemaciclib. And when looking at the patients with an ESR1 mutation, patients did better with imlunestrant versus fulvestrant. We've also been studying imlunestrant in the lab. And one of the things that we first asked is we wanted to assess the selectivity of imlunestrant. And to do that, we looked at the effects of imlunestrant in transcription and looking at what are the transcriptional changes induced by imlunestrant in in hormone-deprived conditions where in wild-type ER cells, there's no active transcriptional effects. As you can see here, when we looked at the differentially expressed genes, really at 6 hours and at 12 hours, there were no transcriptional changes. In contrast, when we looked at the ER mutant cells that have active transcription in hormone-deprived conditions, we did see transcriptional changes with imlunestrant treatment. And we look at what are the transcriptional changes. We saw that the top-ranked pathway downregulated with imlunestrant is are really the canonical ER signaling genes. In addition, when we looked at ER degradation in response to imlunestrant. When we looked at the wild-type ER cells, we didn't see differences compared to fulvestrant. However, when we looked in ER mutant cells, we saw that imlunestrant did induce ER degradation, whereas with fulvestrant there was lower degradation, already showing us evidence of differences between imlunestrant and fulvestrant in the setting of the ER mutations. So to further study these differences, we collaborated with Sean Fanning from the Loyola University, and he did molecular docking of imlunestrant to the Y537S ER mutation mutation LBD to predict the different effects of these two different antagonists. And as you can see here, really looking at this is looking at imlunestrant. You see nicely that imlunestrant sits in the binding pocket. And when we overlay imlunestrant, which is in green, with an ICI or a drug closer to fulvestrant, which is in cyan, you can see clearly that the structures are different and that their interactions with the ligand binding domain and specifically with helix 12 are very different, already giving us evidence that they're doing something very different. And when we look in a PDX model that harbors a Y537S and look at the activity of these drugs, we see clearly that imlunestrant has really more potent tumor suppressive activity. And looking at the transcriptional effects, already at day 10, we see really strong inhibition of proliferation with imlunestrant compared to vehicle control or fulvestrant, and this proliferation inhibition is maintained on day 28 as well. So in addition to investigating oral SERDS and second-line treatment, there are also now a number of phase 3 studies looking at oral SERDS in first-line treatment for metastatic disease in combination with palbociclib. Most of these studies are looking at patients who are considered endocrine sensitive. There's also the Pioneer study that actually has a different design and actually is looking at patients who are endocrine resistant and enriched in patients who have ER mutations. In addition, there are also now clinical trials even looking at oral SERDS even earlier and looking at in the adjuvant setting. So there are now several large phase 3 trials looking at the different oral SERDS in the adjuvant setting. And these studies have different designs, looking at oral SERDS either up front or looking at oral SERDS as extended endocrine therapy, or also looking at the possibility of randomizing patients based on MRD monitoring. So these studies are still ongoing, and we're awaiting their results. So to summarize, SERDS anti-activity is likely due both to its antagonistic activity as well as degrader activity, and I would argue that really SERDS should be called selective estrogen receptor antagonists and degraders, or SERDS. We know that better ER degradation does not necessarily mean a more potent drug, and likely ER degradation by itself is not a sufficient marker to detect better anti-tumor activity. The new oral SERDS have unique properties compared to fulvestrant. They have better bioavailability and PKs. Their chemical structure is different, and their interaction with the ligand binding pocket and helix 12 positioning and coactivator binding is likely also different. We already know that they have a different side effect profiles as well. So what about in the clinic? Where are we heading and what do we know? So as far as right now in metastatic ER-positive breast cancer for second-line treatment, there is an approved single-agent oral SERD, and this is for patients with an ER mutation, and likely, this is really for patients who have shown endocrine sensitivity by showing response to CDK4/6 inhibitors and endocrine therapy at first line for at least 12 months. There are ongoing studies looking at combinations of SERDS with other targeted therapies, and right now it's not clear if this will be for all patients or ER mutant patients alone. In addition, these oral SERDs are now being investigated in combinations with CDK4/6 inhibitors as first line. Unclear if we should be giving these, if these would be better up front, or should we be using, should be doing CT monitoring and then switching based on this? Should this be for patients who have endocrine resistance? And then I would also say that one of the things that we're interested in is understanding if actually we should be continuing to block ER even after transitioning to chemotherapy or other types of treatment, since the ER, particularly in the setting of the ER mutations, which really signifies an ER addiction. In addition, in the early stage setting, there are ongoing large clinical trials to look at the role of SERDs in that setting. And again, one of the questions is, should this be used up front as extended treatment or based on ctDNA monitoring? So really still many questions, and really we need the results from these ongoing trials and future trials, and also studies of biomarkers to really understand who benefits and when do patients benefit from the oral SERDs. So with this, I will end, and like to thank the members of my lab. Also want to thank my collaborators and colleagues that have given me advice for this talk today.
Thank you. Thank you. Questions for Dr. Jeselsohn?
Nick Murray, you've described different structural changes with different SERDs, and you didn't say it, but I took it as an implication that there may be different functional consequences, perhaps different patterns of changing transcription. That would seem to open up a whole set of possibilities for selecting a SERD based on perhaps other characteristics of the tumor. Could you just talk a bit about that?
Yeah, so we don't have that data, so we don't know how. As I showed you, we've done some modeling of imlunestrant, but I'm not aware of crystal structures that have really teased this out on how the different SERDs are really doing it. We just can speculate. Is this really going to translate to differences in the clinic? I don't think we know yet. So it could, but I think I can't answer that. I don't think we have that information.
Yeah, Donald?
Sorry, I think you did a gorgeous job of highlighting obviously the positives of where they come from, but also what the problems are. And I'm actually going to suggest a rather heretical position, is that we've lost our way somewhat, in the sense that at least I think we've become so hyper-focused on degradation, as you pointed out. Degradation doesn't seem to be the answer. The acrylic SERDs, even in the clinic, the first one we developed, Ataxil developed, it was incredibly good, even in the clinic at degrading receptor, but there was virtually no efficacy. And so it comes back to the point of, I think there's something missing. I saw the Ember trial this morning. I was very disappointed because there was no activity in the wild type, very little in the wild type population. I thought we were finally going to get a drug that's going to give us. So I put the question to you, maybe when we're focusing on the cancer cell only as the target, that's great with ESR1 mutant cancers, because it's telling us it's endocrine addicted. I'm not saying obviously the estrogen receptor is not important, but I think it's estrogen receptor extrinsic to the tumor that's impacting the biology of these SERMs and SERDs. And of course, we're not really looking at that right now. And then I'll just finish, because on that note, is saying that the best SERD out there is estradiol.
Is what?
Estradiol. Yeah.
Estradiol actually degrades the receptor better than any of the other SERDs, okay? And actually, estradiol in the head-to-head with, remember the old data, head-to-head with tamoxifen was equally good, but overall survival was much better in the estradiol-treated patients. So I think the whole idea that we need to get absolute quantitative inhibition of estrogen receptor signaling, I'm personally going to take a step back from and figure out what we missed, because it's clear that in these later studies, we're missing something, because we're not getting the efficacy in the wild type expressing tumors.
So I think to answer,
Sorry for
to answer
Sorry.
that, I mean, it's clear that the estrogen receptor, although it is important, it's, first of all, it's not the only pathway, but the other issue is that really all these drugs that were, both endocrine therapies and CDK4/6 inhibitors are causing they're inhibiting cell proliferation, but they're not causing cell death. And I would argue that we need drugs that will cause cell death, and I agree that, looking just at the tumor intrinsic effects is not sufficient, and we need to look at the tumor microenvironment. And I didn't talk about it, but we have done work that is published showing really that by ER activity, we are enhancing actually NF-kappa B signaling and responses to interferon gamma, and also that can enhance antigen presentation. So it does other things as well downstream that we could potentially leverage for better treatments.
Question on the right?
Yes, I'm wondering about the concept of gain of function of the estrogen receptor mutations in terms of altered DNA binding, for example, as well as the interactions with the steroid nuclear receptors, such as androgen receptor and others.
I'm not sure I understand your question, but
Wondering about gain of function roles of the estrogen receptor mutations.
So altered binding sites.
Yeah, so we have done work on that, and we do show that it does bind to different sites, and that is what we say. That's why I mentioned it does have neomorphic properties, and we show that the transcription induced by these neomorphic sites is really enriched in metastases, and in work that we've done in mice, we show that if you really target the ER mutation, first of all, if you express the ER mutation, you get enhanced metastases, and then also if you target the ER mutations, you have very strong regression of metastases, which highlights the importance of targeting these mutations.
Yep. Next question. Last question.
Thank you very much. Alex Pearson from Institute of Cancer Research, London. So obviously the clinical work is already looking to put these new drugs together with existing compounds, CDK4/6 for instance, but I was wondering from your functional work that you alluded to and showed us a bit of, about acquired resistance to these new compounds. Is it, are the existing drugs, the combinations we're already talking about, the right ones? So the question is about acquired resistance with these new SERDs. Is it the same as
Okay, yeah, that's a good question. I didn't have time to talk about that. So there are some genetic mechanisms of resistance. So there are already known ER fusions that have loss of the ligand binding domain. And obviously, if they don't have a ligand binding domain, none of the antagonists that we have will be effective. There are also other secondary mutations. So Nick Turner's group has shown that there's a secondary mutation for patients, for specifically the ER mutations, there's a second mutation that leads to resistance. We haven't really, it's not really yet completely clear how that happens, so still work to understand, and those seem to be, both of those seem to be rare. In the lab, we're seeing that with treatments with some of these SERDs, we're seeing loss of ER, which could be epigenetic, could be, is it methylation or other plasticity? We haven't seen yet like ER deletions in these models. We are finding in some of our sequencing data, there are also other mutations. We're seeing now that there are truncating mutations leading to loss of ER, but those are also rare. So I think those are the kind of things that we're probably going to see more of as we better target ER with these new SERDs.
So your talk reminds me of the analogy of carboplatin and cisplatin being like Coca-Cola and Pepsi-Cola, remember?
Yeah, yeah.
So, and I don't know whether these SERDs are like Coca-Cola or Pepsi-Cola or Coca-Cola and Pepsi-Cola. So, so I think, so there was a question about, structure-based differences of this, or merits, strengths versus not. So what about, there was an irreversible circa.
Yeah.
I don't know what happened to that, but any merit of thinking of an irreversible, would that be biochemically a better way of inhibiting the receptor, or not really?
Yeah, I don't think so. I mean, these are really degrading also, so in some ways you really are losing ER as well. So I don't think that that's going to be much better. The circa, as far as I know, they're not, doesn't seem to be, the development doesn't seem to be ongoing. They had some issues with toxicity. I mean, I think most of these novel these oral SERDs, like mentioned, and again, we don't know exactly all the structures and how they differ. We already have signals that their side effects are different, telling us that they are doing something different, and I think that's going to be a part of how they're, dictate some of these as well. But their activity and bottom line might be very similar at the end of the day, kind of like, we have 3 different aromatase inhibitors, 3 different CDK4/6 inhibitors. Yeah.
This is one situation where the way that drugs are developed clinically may make a difference, right?
Yeah.
Which is also an
Depending
important
on-
part of
Yeah, I mean, and
the
we're seeing
equation.
that
Yeah.
because the clinical trials, the way they're designing them is really changing
Exactly.
how they're getting into the clinic. Yeah.
Fascinating. Well, thank you very much. And with that, I would like to close this session. Thank the speakers for staying on time, and thanks all of you for being engaged and all the questions that you raised. Thank you.